Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to understand what is validation part 2. So in the previous lecture we have seen the overview of what is validation and how it is useful in any pharmaceutical industries, or you can say what is the need of validation in any pharmaceutical industries and how they are related for the quality assurance of the pharmaceutical drugs. So we have also discussed about some of its type. So today we will going to understand types of validation in detail. So as I have already told you that there are basically four types of validation. The first one is analytical method validation, second is your equipment validation, third is your clinic validation, and the last one is the process validation. So we will going to cover all these types in detail one by one. But before understanding all the types of validation, let me give you a brief introduction about what is validation. So as already discussed in the previous lecture that validation it is a process which is in the form of a documented evidence which provides a high degree of assurance to desired result with the predetermined compliances. Okay, so and it is widely used in pharmaceutical industries. So what is the objective behind this use of validation? So the basic objective, or you can say the concept of validation, is to provide a drug that is free from any source of microbial contamination, or you can say a drug that have a high quality, or a drug that follows all the guidelines of GMPs, GLPs, all the regulatory bodies guidelines is, and the drug is, yes, free from any source of contamination, and it is very much useful, or you can say it is very much safe for the human consumption. Okay, so this is the whole scenario behind the validation. And we have also discussed that this concept of validation was first proposed by FAD, that is a Food and a Drug Administration, in 1970s. Okay, so basically it improves the quality of the pharmaceutical product. Simple. If anyone asks you what is the concept, or what is an objective of validation, so the basic objective and the basic concept of validation is to improve the quality of pharmaceutical product. Okay, and since a wide varieties of procedure and a method, or you can say the activities, are validated to check and improve the quality of the pharmaceutical product. Now coming to the main part of the topic, that is the type of validation. So students, the first type of validation is the analytical method. The second one is your equipment validation. Third is your cleaning validation. And the last one is the process validation. Okay, so whenever it comes to an analytical types of validation, so this type of validation, student, okay, so this type of validation is basically the purpose of analytical validation is just to verify that the selected analytical procedure will give a reliable result that are adequate and are intended for the purposes. Means basically this method is used to verify that all those method or all those procedure that are used in the manufacturing of any type of pharmaceutical drug is completely completely free from any source of contamination. It's completely safe, and the tests that are carried out gives the correct result, or you can say the desired result. So methods should be validated. So basically why there is a need of the method validation, or why the methods are validated? Okay, so the first need is when they are to be established for a routine use. Okay, whenever the method is used, or whenever they are established for the routine use, then it is a necessity to validate that method. Second point says when the method is to be changed due to the change in the condition. If there is a change in any condition and you want to switch to a new method, so it is very much useful, or it is you can say ये चीज़ कही जाती है कि जो नया method है, that new method should be validated just to check whether by using this method the result would be of the desired result or not, or you can say whether the quality is of the product is maintained or not, will be maintained or not by using this particular method. Because whenever anything that is newly used in any of the companies, it is the duty of the employees of the companies to check the method or to check the new technologies that is going to be used. Okay, just to make a safe move, so that the product means देयर कुड मतलब कि आपके प्रोडक्ट के साथ कुछ ऑल्टरेशन ना हो जाए, और प्रोडक्ट शुड बी सेफ. ओके, सो फॉर assuring the quality of the product, it is very much useful to validate the new method. Okay, now the third point says that whenever the equivalence between a new method and the standards are demonstrated, then also analytical method is then also the method is validated. So there are different types of procedure to be validated, or you can say there are different types of parameters to be validated. The first parameter is your accuracy, then is your precision, then it comes that repeatability, then reproducibility is there, then linearity is there, then the range is there, detection limit is there, quantitation is there, quantitation is there. So why there is a need or of these parameters to be validated, we will going to cover one by one. Okay, so before understanding this, let me give you one thing, that it is something that the basic concept behind every validation method is to make a product that will give you a desired result, a product that is that passes all the guidelines. Okay, so the first one is your accuracy. So the accuracy of an analytical method, it basically refers to the closeness of agreement between what observed value and the value which is either conventionally a true one or a reference one. So what is accuracy? Accuracy means the correct thing, accurate thing of any product. Okay, so it is very much important to check the closeness of agreement, that is between the observed value and the values which is either you can say conventionally a true one or the reference value. There's a two values now, the reference value and the true values. Okay, if you don't know the true values and the references value, you can just go to the Google and just search for these two, and in the next lecture I'll be again if any doubt I'll be explaining these two things. Okay, so accuracy, then precision. So the precision of an analytical method is basically refers to the closeness of the values obtained from a series of tests. Whenever you are performing a series of test, right, so it might have different types of values. So then there is a need of validation just to check which values is correct or not. Then repeatability. So repeatability is basically the establishment when the same sample is estimated, or you can say is repeatedly by the same analyst using the same analytical method within a same laboratories using same instrument and perform within a short period of time. So repeatability, since the name suggests repeat, मतलब कोई चीज़ repeat हो रही है यहाँ पर, अगर same method is repeated, or you can say the same equipment is repeated, or the same laboratory is repeated, means everything is same. Okay, so there is also this validate method is to be validated, okay, just to check the you know accuracy or of the product. Okay, or you can say the accuracy is an not, you can say not the accuracy is the right word, but to check whether the method that is again and again is used gives the correct or the result or not. Okay, now let's move further and let's see what is intermediate precision. So when a test is repeated on different days, okay, not on the same days, but here that different days are chosen, and on that particular dates the test is performed, and here the persons are different, and he is using different instrument within the same laboratory, so variation is expressed in terms of intermediate precision. Okay, now comes the reproducibility. Okay, here one thing I would like to mention here, that in examination they can ask you about what is the difference between this intermediate precision and repeatability. Okay, so this is very important and you must be knowing the difference between these two. Then comes the reproducibility. So when the method is standardized, okay, whenever the method is standardized, so the test is carried out in different laboratories. Now here laboratories are changed, means different types of laboratories are used, and using the same method is same, but the laboratories is different. So precision between the laboratories is refers to as a reproducibility. Okay, then comes the specificity. Specificity means specific. Okay, when the specificity is you can also say that it is the ability of a test method to measure the analyte explicitly in the presence of other components. Okay, this is specificity. Now comes the next, that is equipment validation. Okay, so now let's understand what is a equipment validation. So equipment validation is established, you can say documented setup that proves that an equipment that is being used in the manufacturing of a particular drug, it works correctly, and it leads to accepted and accurate result, means whatever a predetermined result is that, so it is giving that result. Means the process of equipment validation is basically based, you can say, on the principle that the equipment must be designed, first thing, okay, designed, constructed, second, maintained, and you can say adapted to perform the operation which are carried out. Okay, so here the equipments, they are the basic component of pharmaceutical industries. We all are aware of that, this thing, that it is the basic component, basic need of any pharmaceutical industries. भाई equipment अगर होंगे तभी तो कोई चीज़ आप बना सकते हो, राइट? So therefore, before performing a process in any pharmaceutical industry, it is become a primarily, you can say, important to issue, or you can say to issue equipment validation. Okay, just to check the quality of the equipment, whether the equipment that you are going to be use in the manufacturing of any particular drug gives the adequate result or not, or accurate result or not. Okay, so here it is written that equipment validation is to provide a high level of documented evidence that the equipment and the process conformed to a standard. Okay, now comes that there are different types of equipment validation. For example, there is a installation qualification, that is IQ. Then there is a operational qualification, that is OQ, design qualification DQ, and the last one is your performance qualification PQ. Okay, but here you have to just understand these what is equipment validation. Okay, so moving to the next, that is the clinic validation. Okay, one thing I would like to mention here is that, just give me a second. one thing, I want everyone to go through what I'm just writing it here for the better understanding that what are the process of validation, equipment validation is mainly divided into the three, sorry. Okay, so here I'm just writing that there are three phases of validation. Okay, what are the three phases of validation? So this is the thing I would like, I haven't mentioned in the ppts, but you must be aware that there is basically three phases of validation when it comes to the equipment validation. Okay, the first one is the pre-validation phase, second one is your process validation phase, and the third one is your validation maintenance phase. So I would like everyone to go through this three phases. I'm just again writing all the three phases. You can take a screenshot if you want. The first phase, okay, the first phase, that is phase 1, that is the pre validation phase. Okay, this is the first phase. Okay, pre-validation phase, basically here you check the design qualification comes under this. And the next, you can say, the next phase is phase 2, that is process validation phase. Okay, and the last one is validation, I'm just write validation maintenance phase. Okay, so these are the three phases. The first is a pre-validation phase, second is your process validation, and the last one is the validation maintenance phase. So you must be aware of all these type phases that is there in equipment validation. Okay, now moving further, and let us understand what is the cleaning validation. Okay, so cleaning validation, basically it involves, or you can say it assures that there is no cross contamination in multi product manufacturing plant, and also prevent microbial contamination. So since the name suggests cleaning, so the basic aim behind the cleaning validation is to check that there is no such cross contamination between in the manufacturing plant, and everything that is manufactured in this manufacturing plant is safe and it is free from any source of microbial contamination. So this is the basic aim behind the cleaning validation. Okay, so the cleaner, once the product is manufactured, the equipment is cleaned using a appropriate cleaning SOPs. Okay, so there are some appropriate cleaning SOPs are there, so you should always use those SOPs to clean the equipment. Okay, just to check, just to remove all the residues that is there. And so this is the cleaning validation, and there are different types of contamination to be considered in cleaning validation. So while you are cleaning in the equipment or sanitizing the place, two or three things should be keep in mind while doing the cleaning validation. The first one is the cross contamination. Okay, second is your microbial contamination, then is your contamination by cleaning or sanitizing agent, and the last one is contamination by other agent. So these types of contamination you should always consider in cleaning validation. Okay, so this is the easiest type of the validation. And now comes the most important types of or you can say method of validation, or you can say types of validation. That is the process validation. So this is very important, student. That process validation, it means or assuring and providing documentary evidences that are capable of repeatedly and reliably producing a finished product of the required quality. Okay, so what is the process validation? So process validation is one of the most important type or method of validation. And it is a step by step procedure, where is a sorry a process validation, you can say, is a step by step procedure designed to assure that the manufacturing process can be consistently produce a quality product. Second, it is performed by validation team lead by the quality assurance head of manufacturer, and that is there in the pharmaceutical industry. So basically you can say that the quality that the process validation is done before releasing a new product. Okay, whenever you want to release a new product into the market, so the first step is to do the process validation. Okay, so what are the processes that you are going to use? Okay, so this is one of the most important types of validation. Okay, so under the process validation there are different types, like prospective validation, then is your concurrent validation, then is your retrospecific validation, and then is your process revalidation. Okay, so in process validation also there are the things that you should always keep in mind, that what is a process design, means how you are designing a process. Okay, what are the design of the processes that you are going to use. Then what are the process qualifications? Qualification comes means like premises, your utilities, your equipment, your commercials, scale batches. Okay, so that these all comes under the process qualification. How many number, or how many batches, or you can say the number of batches that you are going to be used. Then is your continued process validation, that a periodic review of the trends may be, it includes the sampling and test inline, online, or you can say at line monitoring is also very important. And in a process validation, or in yes in sorry in a process validation, it is very much important that what two or three things has to be checked. That is risk management, how to manage the risk. Okay, how to change the control if there is a sudden change. Okay, so how you should control that change, or how you maintain the product life cycle. So these thing comes under the process validation. Okay, now moving to the process, just let me give you a brief example, just to get more knowledge of validation, means how validation of any solid dosage form take place in the laboratory. तो जितना अभी हमने पढ़ा, अब हम समझेंगे कि अगर एक सॉलिड डोसेज फॉर्म है किसी भी मेडिसिन का, तो उसके ऊपर वैलिडेशन कैसे किया जाता है. ओके, so here the objective of the design and manufacturing of solid dosage form, that is capsule or tablets. ठीक है, tablets हुआ, capsule हुआ, they comes in the solid dosage form. So it is basically deliver orally and the correct amount of drug in a proper form over a period of time and the desired location, and have its chemical integrity protected to that point. Oh yeah, so whenever you are taking any type of a solid drug, so you take that drug under the supervision of your doctor. Okay, so you take the drug in the proper form, you can say proper dosage form, and over a period of a proper period of time. Okay, so the prime objective of any pharmaceutical plant or industry is to manufacture a product of high quality. obviously every industry needs a product to be manufactured of a high quality, because if the quality is high, there is a repeat customer, and the revenue of the company will be boost up. Okay, if the quality is wrong, if the quality is very low, nobody will going to purchase that particular product, and your cost also will be ruined. So it is very much important for any pharmaceutical industries or any food industry or any beverage industries to manufacture a product that is of a high quality product. Okay, so there through a careful design, validation of both the process and the controls, manufacturer can establish a very high degree of confidence that all the manufactured unit from a successive lots will be acceptable. Okay, so these things should be kept in mind whenever you are buy any pharmaceutical plants. Okay, the product quality can be assured by, नाउ अगर अब आपका प्रोडक्ट इज मैन्युफैक्चर्ड, अब उस प्रोडक्ट की क्वालिटी को कैसे एश्योर करोगे? द फर्स्ट इज बाय द फिजिकल एंड द केमिकल स्टेबिलिटी, यू चेक द केमिकल एंड द फिजिकल स्टेबिलिटी ऑफ दैट पर्टिकुलर प्रोडक्ट. Okay, now at second is the preservation against microbial contamination. Now what are the preservative or preservation techniques you will use just to protect the product from the microbial contamination? Because you all know that microbial contamination is one of the contamination that can degrade the quality of the product. So it is very much important that a specific preservative, or you can say accurate preservative techniques should be used to avoid the microbial contamination. Then comes the uniformity of the dose of the drug. Okay, then is your acceptability of the users, means how the user are using or accepting that particular drug is one of the importance. Then is the suitable packaging and labeling. Okay, then is the last is your validation. Hmm. Then the tablets that you are using in the as a dosage form comprises a mixture of active substances and excipient. Okay, so basically they are usually in the powder form or pressed compacted in the solid. Then the excipient includes the blenders, glidants, lubricants to assure the efficiency of tableting, disintegrant to promote the tablet break up into the GIT. So these things should be kept in mind while doing a validation of any solid dosage form. Same cases are with the liquid also, whenever there is a medicine that is in the liquid form. So this brings to the end of the lecture. I hope you have understood the lecture. So if you have any doubt, any queries related to the lecture, you can simply leave your queries in the comment section below. In the next lecture, I'll be answering the queries. Follow ITLS Academy on different portal. You can now visit our website, that is www.itlsacademy.com. This is our helpline number, that is 7080833450. You can simply call on this number and get updated. So thank you for watching. Please do like, share and subscribe. And thank you once again for watching. Thank you.